Hi student, in this video I am going to talk about the enzyme inhibition. Before starting this video, I would request to all of you, please share this video and please subscribe our channel and please press the bell icon for get more updates from our channel and please like our videos. So let's start today's topic, enzyme inhibition. Inhibitions of enzyme activity may be irreversible or reversible. In irreversible inhibition, inhibitor called irreversible inhibitor is binds tightly to the enzyme either covalently or non-covalently. An inhibitor dissociates very slowly from the enzyme and enzyme's catalytic activity is permanently inhibited. The antibiotic penicillin acts as an irreversible inhibitor of the enzyme glycopeptide transpeptidase. This glycopeptide transpeptidase, also known as glycoprotein peptidase, and penicillin covalently bind with an essential serine residue in the active site of glycopeptide transpeptidase, an enzyme that acts as cross-link the peptidoglycan chain during the synthesis of bacterial cell walls. Once the cell wall synthesis is blocked, the bacterial cells undergo osmotic lysis and bacterial growth is halted. And similarly, aspirin binds covalently with the enzyme cyclooxygenase, reducing the synthesis of prostaglandin. And another inhibitor, that is suicide inhibitor, I also describe in briefly in later. Suicide inhibitor, this is the mechanism based inhibitor, is a type of irreversible inhibitor, and suicide inhibitor binds to the enzyme as a substrate and is initially processed by normal catalytic mechanism. As a result of catalysis, substrate is converted to chemically reactive intermediate that inactivates the enzyme through covalent modification. That is, enzyme literally commits suicide. In reversible inhibition, inhibitor called reversible inhibitor binds non-covalently to the enzyme and dissociates rapidly from the enzyme. The effect of reversible inhibitor is reversed after dissociation of inhibitor from enzyme, and there are three common types of reversible inhibition. These are competitive, uncompetitive, and non-competitive inhibition. Competitive inhibitions or reversible inhibitions. One of common type of reversible inhibition is called competitive inhibition. A competitive inhibitor competes with the substrate for an active site of an enzyme, while the inhibitor, that is represented by capital I, occupies the active site. It prevents binding of the substrate to the enzyme. Many competitive inhibitors are compounds that resemble the substrate and combine with the enzyme to form an enzyme inhibitor complex, that is EI complex, but without leading to catalysis. Even fleeting combinations of this type will reduce the efficiency of the enzyme. By taking into account the molecular geometry of inhibitor that resemble the substrate, we can reach conclusions about which parts of the normal substrate bind to the enzyme. Competitive inhibition can be analyzed quantitatively by steady-state kinetics. In the presence of a competitive inhibitor, the Michaelis and Menten equations becomes V0 is equal to Vmax into concentration of substrate by alpha Km plus substrate concentration, where alpha is equal to 1 plus concentration of inhibitor by K suffix I. and K suffix I, that means K suffix I is equal to E into I, concentration of enzyme, and multiply concentration of inhibitor by concentration of EI complex. And this equation describes the important features of this equation means the Michaelis-Menten equation of competitive inhibitor. Important features of competitive inhibition: this experimentally determined variable of alpha Km, and the Km observed in the presence of the inhibitor is often called the afferent Km. Because of the inhibitor binds reversibly to the enzyme, the competitions can be biased to favor the substrate simply by adding more substrate. When substrate for exceed, and there is a very common question which comes in examination, that is, with competitive inhibitor, the reaction can eventually reach its normal Vmax, but it takes higher concentration of substrate to get it there. And in other words, Vmax is unchanged, but the apparent Km is higher. And the question is, why must more substrate be added in order to reach Vmax? The extra substrate makes the substrate molecules abundant enough to consistently beat the inhibitor molecules to the enzyme. And this is the answer of this question. And this, when substrate concentration far exceed the concentration of I, the probability that the inhibitor molecules will bind with the enzyme is minimized, and the reaction exhibits a normal Vmax. However, the substrate concentration at which where the V0 is equal to half Vmax, then the apparent Km increases in the presence of inhibitor by the factor of alpha, and this effect on afferent Km combined with the absence of an effect on Vmax is diagnostic of competitive inhibition and is readily revealed in double reciprocal plot. And this is the double reciprocal form of equation. And the equilibrium constant for the inhibitor binding, that is K suffix I. I discussed in previous what is the K suffix I. This is the equilibrium constant for inhibitor binding, can be obtained from the same plot. As I told, in competitive inhibitions, Vmax stays same and Km increases, but the inhibitor does not affect the turnover number of the enzyme. Clinical treatment of methanol poisoning is a classical example of the exploitations of competitive inhibitory mechanism. In the case of methanol poisoning, methanol in the body is converted to harmful formaldehyde by alcohol dehydrogenase. A high dose of ethanol is used to alleviate the effect of methanol because ethanol competitively binds with the active side of the alcohol dehydrogenase. Similarly, methotrexate is a potent competitive inhibitor of the enzyme dihydrofolate reductase, which plays a role in biosynthesis of purines and pyrimidines. Methotrexate acts as a structural analog of dihydrofolate, a substrate for dihydrofolate reductase. Two other types of reversible inhibitions, that is uncompetitive and mixed, though often defined in terms of one substrate enzyme, are in practice observed only with enzymes having two or more substrates. An uncompetitive inhibitor binds at the site distinct from of substrate active site, and unlike a competitive inhibitor binds only the enzyme substrate complex. In the presence of an uncompetitive inhibitor, the Michel's-Menten equations is altered to V0 is equal to Vmax multiply the concentration of substrate by Km plus alpha dash multiply substrate concentration, where alpha dash is equal to 1 plus inhibitor's concentration by K dash suffix I, and K dash suffix I is equal to Es. K suffix I dash is equal to Es multiply I, the concentration of Es multiply concentration of inhibitor concentration of ESI complex. As described by these equations, the Michel and Menten equations of uncompetitive inhibitor at high concentrations of substrate, V0 approaches Vmax by alpha dash. Thus, an uncompetitive inhibitor lowers the measured Vmax. Apparent Km also decreases because the substrate required to reach 1/2 Vmax decreases by the factor alpha dash. And the double reciprocal form of the equation will be in the uncompetitive inhibitions 1 by V is equal to Km by Vmax multiply 1 by substrate concentration plus alpha dash by Vmax. And mixed inhibitor also binds at the sites distinct from the substrate active site, but it binds to either enzyme or enzyme substrate complex. The rate equation describing mixed inhibition is V0 is equal to Vmax Vmax into substrate concentration by alpha Km plus alpha dash substrate concentration. A mixed inhibitor usually affects both Km and Vmax. The special case of alpha is equal to alpha dash, rarely encountered in experiments, classically has been defined as non-competitive inhibitions. Examine the equations of the Michaelis-Menten equation of mixed inhibitor. To see why a non-competitive inhibitor would affect the Vmax but not the Km. This equation serves as a general expression for the effects of reversible inhibition. And pure non-competitive inhibition occurs if Ki is equal to Ki dash. In this type of inhibition, Vmax decreases and Km stays constant. And the double reciprocal form of the equation will be 1 by V is equal to alpha Km by Vmax multiply 1 by substrate concentrations plus alpha Vmax. Now I am talking about the irreversible inhibition. And the irreversible inhibitor are those that bind covalently with or destroy a functional group of an enzyme that is essential for the enzyme's activity, or those that form a particularly stable non-covalent association. Formation of a covalent link between an irreversible inhibitor and an enzyme is common. Irreversible inhibitors are another useful tool for studying reaction mechanism. Amino acid with key catalytic function in the active site can sometimes be identified by determining which residue is covalently linked to an inhibitor after the enzyme is inactivated. And the example is a special class of irreversible inhibitors is the suicide inactivators or suicide inhibition. This compound are relatively unreactive until they bind to an active site of a specific enzyme. A suicide inactivator undergoes the first few chemical steps of the normal enzymatic reaction, but instead of being transformed into normal product, the inactivator is converted to very reactions compound that combines the irreversibly with the enzyme. And this compound are also called mechanism based inactivator because they hijack the normal enzyme reaction mechanism to inactivate the enzyme. Suicide inactivators plays a significant role in rational drugs design, a modern approach to obtaining new pharmaceutical agents in which chemists synthesize novel substrate based on knowledge of the substrate and the reaction mechanism. A well-designed suicide inactivator is very specific for a single enzyme and is unreactive until within an enzyme's active site. So drugs based on this approach can offer the important advantage of few effects. And reaction of chymotrypsin with diisopropyl fluorophosphate DIFP irreversibly inhibits the enzyme, and this has led to conclusions that serine 195 is the key active site of serine residue in chymotrypsin. And another example, suicide inhibitor of Mono-Oxime oxidase for treatment of depression. Here we know that norepinephrine and serotonin are the neurotransmitter, and this norepinephrine and serotonin are normally destroyed by an enzyme that is monoamine oxidase. This monoamine oxidase, if we use some inhibitor, monoamine oxidase inhibitor blocks this enzyme, that is MAO, and this inhibiting the destructions of non-epinephrine and serotonin and allowing the neurotransmitter to remain active longer, and it used in the depression treatment. In this video, I discussed the inhibitions of the enzyme activity. If we summarized, reversible inhibition of an enzyme is competitive, uncompetitive, or mixed or non-competitive inhibition. Competitive inhibitors compete with the substrate by binding reversibly to the active site, but they are not transformed from the enzyme. In uncompetitive inhibitor, bind only to the ES complex at the site distinct from the active site, and mixed inhibitor, or you may say non-competitive inhibitor, binds to either enzyme or enzyme substrate again at the site distinct from the active site. And in irreversible inhibition, an inhibitor binds permanently to an active site by forming a covalent bond or a very stable covalent interaction. From this video we can say competitive inhibitor is Km-positive inhibitor. Their Km increases but Vmax does not change, and non-competitive inhibitor, that is non-Km-positive inhibitor, their Km does not change but Vmax decreases. I hope this video will help you to understand the inhibitions of enzyme activity. If this video will help you, please share this video to among students, and please tell them please subscribe our channel and press the bell icon to get more updates from our channel. And please don't forget to like our channel. Thank you from Knowledge of Life Edu.